Hi there, welcome to my lecture on marsupial reproduction. So the objective of today's lecture is to provide an overview of marsupial reproduction and consider some marsupial strategies and specialties in relation to both reproductive form and function. So from the information provided in this lecture and from your own reading, you should gain an understanding of the reproductive anatomy of male and female marsupials, acquire knowledge of early development in marsupials, and understand that it is a specialist arid adapted strategy. And you should appreciate the differences and similarities between marsupial and placental reproduction. So why is reproduction important? Well, sustaining populations requires a good understanding of the environmental and biological factors that ensure species survival. And obviously reproduction is going to be key to this this information and to ensure the survival of the species. So studying the reproductive biology of wild animals produces knowledge beneficial to their management and conservation. As you know, many Australian marsupials are threatened, or probably most of them are. So understanding reproduction is going to be critical to efforts that will ensure their survival, as well as enhancing breeding success in ex situ populations. Understanding reproduction and it's important, and as an important factor, is also a prerequisite to the development of assisted reproductive technologies. And this can have a huge outcome for overcoming mating difficulties and addressing infertility issues and maintaining genetic diversity in rare and endangered populations. So just a overview of mammalian reproduction broadly. We know that monotremes are a very early diverging group from other mammals, and that marsupials and placentals actually diverge much later on in evolutionary history. So these three extant lineages of mammals are actually distinguished primarily based on their reproductive strategies. So monotremes, what is it about monotremes that makes them different from marsupials and eutherians? And it basically comes down to the separation of the digestive, urinary and reproductive tracts. Well, there's differences in the separation of the digestive, urinary and reproductive tracts that distinguish the three groups, three mammal groups. So female monotremes, and monotreme actually means single hole, they have a single opening for excreting digestive and urinary waste and for reproduction. So this hole is referred to as the cloaca. They lay eggs from which altricial young hatch. So altricial young is very underdeveloped and dependent young. And monotremes also lack nipples, which is interesting. So young, instead of attaching to nipples, they actually suck secreted milk from the fur of the mothers that is produced by glands under the skin or within the skin. So placental or eutherian mammals, obviously we know that they have a complete separation of the digestive, urinary and reproductive tracts. They have a vascularized placenta that will support the embryo through development, and that entailing a long digestion period. So they have a single vagina for birth. In comparison, female marsupials have a urogenital sinus. So this is separate from the digestive tract, but the waste of the bladder and the purpose of reproduction does occur through this one opening, which is the urogenital sinus. Marsupials have paired vaginae and two cervixes, and sperm will traverse to the uterus through these paired lateral vaginae. They have a pseudovaginal canal, which is for birthing young, and they have a transient placenta. So this is formed by the chorion and the yolk sac. And then obviously we know that they also have a pouch, and this is where the extremely altricial young develop. So today's lecture, we will focus on certain aspects of marsupial reproduction within the whole kind of reproductive life cycle of a marsupial, because there's obviously many different elements to reproduction. So this lecture will focus on insemination to independence of the young and will include reproductive structure and function, and we will cover some fascinating reproductive strategies. We are not dealing with the reproductive hormonal control side of reproduction. So if you want to learn about that, there is plenty of literature out there for you to read up on. And then next lecture, we will talk about sexual selection, which is obviously another part of reproduction. And sexual selection also involves elements of things that we've already covered in communication, but we will get to that next time. So starting with anatomy, male reproductive anatomy of marsupials are similar to placental males in that they consist of the testes, which is where sperm are produced. They have a specialised duct to transport the sperm to the external environment, and they also have secretory glands that produce the seminal fluid. So testes are held inside an external scrotum in most mammals. There are some major differences, though, that we do see in marsupials. So you should know by now from your spotting of the testes that marsupial testes are positioned in front of the penis and not behind it. And there is a difference in the relationship between the positions of the uterus and male ducts between male marsupials and male placental mammals. So the testes of marsupials, they're basically paired oval structures that are surrounded by the tunica vaginalis, and this is actually usually quite characteristically black due to its pigmentation. So the testes of marsupials tend to be pendulous. The scrotums of marsupials tend to be pendulous, and this allows cooling by radiation. And the testes are typically 3 to 4 degrees cooler than the body temperature. This is also cooling of the testes is assisted by a specialised muscle that will retract the testes against the body in warmer weather, not cooler weather. So fur is distinctively patchy in the testis region, and there's also very little fur covering the epididymis, and this again is to maximise cooling of that area. There are some species that lack a pendulous scrotum, marsupial moles, which have internal testes, which makes sense due to their subterranean existence. A pendulous scrotum wouldn't work so well burrowing underground. And wombats tend to have a scrotum that is quite small and held close to the body. Sorry, that's specifically southern hairy nose wombats. But weirdly, in the other wombat species, we don't see this. So that's interesting. It may have something to do with climate restrictions or climate limitations. So sperm production, like all mammals, the male testes contain seminiferous tubules, and this is where the sperm are produced. Sperm move from the testes. So, sorry, just to quickly go through how sperm are produced in the testes. If you look at the figure, you'll see the seminiferous tubules. which are surrounded by interstitial tissue. So this is a cross-section of the testes. And it's those seminiferous tubules that produce the sperm. So the sperm are produced and then pushed out into the tubule lumen to be passed down through to the epididymis. And this epididymis will then have has three different regions. So the caput epididymis, this is the first region that the sperm are found in the epididymis, and they become more concentrated here. They then move along the corpus epididymis, so that's sort of the region that kind of branches on the outside of the testis, and this is where the sperm undergo maturation. And so from moving from that branched corpus epididymis along the outside of the testes, they then reach the caudal or cauda epididymis for storage until ejaculation. So sperm are then carried from the epididymis via the vas deferens, and this will actually join up at the urethra just below the bladder. So when sperm are being ejaculated, this is caused by muscular contractions of the epididymis and the vas deferens, and then those larger numbers of stored sperm in the caudal epididymis will pass from there into the urethra for insemination in the female. And so some sperm will pass down the urethra and be voided in the urine, and this usually occurs when the males are becoming ready to or coming into breeding condition. And this is often used in ex situ populations to assess male breeding status, so when they're ready to go to be paired with a female. The sperm of marsupials are very similar to other mammal groups in many ways. So they consist of a small head which contains the DNA, and the DNA will be contained in a nucleus and then have an acrosome present as well. The midpiece region of the sperm contains the mitochondria, and this is what powers the sperm's movement. And then the sperm have a tail, which is basically a single flagellum that starts at the base of the head and down to the very end of the sperm. So the tail does typically kind of include that mid-piece region that we spoke of just before. So the acrosome is a little bit different in marsupials. So in placentals, you can see there in that figure at the top that it is wrapped around the apical region of the sperm nucleus, whereas in marsupials, the head is actually flattened and the acrosome lies to one side. So hopefully you can see my arrow, and that's that region here, whereas this is the acrosome region in a placental mammal. This point I've indicated here as well is a characteristic of marsupial sperm. So this is kind of a specific groove that we see where the mid-piece joins the head of the sperm. So that's unique to marsupials as well. And there's just this interesting fact about South American species. It's known that sperm will pair up during epididymal transit by sort of opposing their acrosomal faces of the heads. And then they are ejaculated in this way, but will separate during transit within the female reproductive tract. So this adaptive advantage is unknown, but it's thought it may be something to do with facilitating directional movement toward the egg, which would have been selected by natural selection, or perhaps it may be important in sperm competition. So it could be that sexual selection is actually driving this paired upwardness of opossum sperm. So male accessory glands, this In marsupials, this includes the prostate gland and the urethral glands, of which they have 1 to 3 pairs. So these glands will produce the bulk of the seminal fluid. Missing in marsupials are the ampullae, the seminal vesicles, and the coagulating glands. So these are a complex of glands that you do find in placental mammals, but not in marsupials. So in marsupials, however, the prostate is proportionally larger relative to what we see in placental mammals. And the prostate gland does change size quite significantly in some species. So in the Tamar wallaby, the prostate, as well as the bulbourethral glands, will enlarge, but the testes actually do not. And this is a little bit different to what we see in other species. So, for example, in the brush-tailed possum, we do see quite significant changes in testis weight, which you can see here up there. But there's also, again, kind of this really significant change in size of the prostate gland. So the penis, marsupials have a bifurcated penis, which means it's separated into 2 columns. They're used only during copulation, and the penis is separate from the urinary tract. When it's not being used, it is retracted into the body in an S shape. The marsupials don't have a baculum, so a baculum is a small bone found in the gland's penis or the tip of the penis. So many eutherian species do have a baculum, but some don't. And so the evolution of that bone is quite enigmatic, and there's been quite a lot of research done on that, including one of my students. The shape of the gland's penis does vary among species, and so in many dasyurid species, penis morphology is actually used as a distinguishing characteristic. And so here you just have an image of the sugar glider penis that has been pulled out from the body cavity. So you can just see that bifurcated form there. So in terms of penis musculature, again, marsupials' morphology is quite different. So in placental mammals, they have these paired muscles that will anchor the penis to the pelvis. So musculus, whatever that word says, I'm not going to attempt to say it, but that is the name of the muscle that anchors the penis to the pelvis. And then placental mammals also have these paired muscles that will converge to envelope the base of the penis. So here is that muscle that pairs that anchors the penis to the pelvis, and here is the one that tends to envelope it. So this is the musculature of the penis musculature in a dog. And so the penis is here, and my understanding is that then the end of the penis is kind of curling up a bit. So it's sort of like an underview there. Marsupials have both sets of these paired muscles, but their anatomy is dramatically different. So they actually remain completely separate. So there's no convergence of those pairs in any way, and they are bulbous and globular in shape. So you can see that here, and contrast this shape to this shape, and vice versa. With in placentals, this is the muscle that anchors the penis to the pelvis. So in marsupials, we do not have this sort of level of connection to the pelvis. So why is this? Well, these muscles function during erection and ejaculation via contractions. So they're involved in increasing blood pressure within the vascular tissue. This thick muscular anatomy of marsupials would definitely be well suited to this function. But this sort of weird absence of any connection of to the pelvis in marsupials suggests that there is a possibility of different mechanisms relative to what's going on in placentals. And it's been suggested that it might have something to do with variation in how blood is returned to the heart, differences in how the penis is averted from the male body, and variations in penile movements such as flips, which apparently is a rapid dorsoflexion of an erect penis. So I think, in summary, this different penis musculature may be related to different ways that the penis is vascularized during copulation and the way it is used during copulation. So moving on now to female reproductive anatomy, we've touched on this a little bit at the start of the lecture, but the system basically consists of an ovary which produces eggs and reproductive hormones. So the hormones that it produces is estrogen and progesterone. And then there's a duct system that leads from the ovary to the outside. So this duct system has specialised functions. It obviously is involved in passage of eggs that are released from the ovary and need to pass down through to the oviduct. It consists of the uterus, which is a specialised space for embryo development. It has a uterus opening, which leads to this kind of really complex vaginal structure that we see in marsupials. And then finally at the end, there is the urogenital sinus. So the ovary structure is similar to that of placental mammals. The ovary cortex will contain numerous egg cells that are held in a resting phase, and then that each follicle and then a follicle which contains an egg cell is then surrounded by a layer of granulosa cells, which again is surrounded by an outer layer of theca cells. So at ovulation, the wall of one or more of these mature follicles will rupture and that will release the eggs. The remaining follicle cells undergo a transformation to become a spherical body, which we refer to as the corpus luteum. So the corpus luteum actually excretes estrogens and progesterone, and it's progesterone that will actually cause hormone changes in the uterus that will make it a suitable environment for the fertilized egg and that kind of some early nourishment of the embryo. So obviously there isn't prolonged placental nourishment of embryos in marsupials, but there is a small duration where they are nourished in the uterus. So if an egg is not fertilized, the corpus luteum will then become inactive and regress. So that rupture site of the ovum in the ovary does have a very important role once the egg is released. So after ovulation, eggs will be collected by the fimbrium, which is a thin membrane that forms a funnel basically at the top of the ovary. And then the egg will pass down to the oviduct, which is a muscular tube, and it's lined by a secretory epithelium, and it functions to kind of coat that egg in glycoproteins as it transits down. In marsupials, there seems to be quite rapid passage of the egg through the oviduct relative to placental mammals. So this happens in about 24 hours from sort of ovulation to reaching the uterus. So the uteri have a muscular exterior layer and an internal secretory layer, which is the endometrium. And this endometrium has a fibrous stroma with tubular secretory glands. So stroma are the cells and tissues that support and give structure to organs, glands, or other tissues in the body. And stroma is mostly made up of connective tissue, blood vessels, lymphatic cells, and lymphatic vessels and nerves. And the endometrium also has a secretory epithelium that again is involved in secreting hormones during gestation and supporting the development of the embryo. So the major differences, marsupials have two uteri with two separate cervixes, and they possess an anterior vaginal expansion, which you can see here. So this is the vaginal cul-de-sac. They have three separate vaginae, so two lateral ones down the side here, and then one median one. So the lateral vagina are for sperm transit, and then the median vagina is for birth. And there's a common passage at the bottom, which is the urogenital sinus, and that is for both reproductive urinary as well as excretory systems to the exterior. So this kind of very dramatic difference in marsupial and placental structure actually arises from a small difference during embryonic development, which I'm not going to go into today, but if you are interested, definitely read up on that and the way that small event does end up with very different reproductive systems. So the basic morphology of female reproductive tracts across marsupials is relatively uniformed. There's some interspecific variation in size and shape of some elements. So this, you also see within species variation and within individual variation in size and shape of the vagina due to variation in reproductive status. So the vaginae and the urogenital sinus can actually grow several fold leading up to the days leading up to ovulation. And the median vagina, in most species, it actually forms as a transient birth canal. So you'll open up just before birth and then close soon after. But in macropodids and the honey possum, it opens up for the first birth and then remains open for the rest of the female's life. So some variation there in the way that the median vagina functions. So females of some marsupials have evolved sperm storage crypts in the uterus or oviducts. So in Antechinus species, and you may remember that this species exhibits seminal parity. So this is when mating, a reproductive event, is followed by the death of all the males in the population. So in these species, we know that there is sperm that are held in oviductal crypts for a short period prior to ovulation. And then in fat-tailed dunnarts, we know that after mating, sperm are restricted to special storage crypts in the oviduct. And it's after ovulation that the sperm in these storage crypts will then migrate to the egg for fertilization. So this here, this letter B, this letter B reflects the point in the oviduct where those sperm are held in fat-tailed dunnarts. Now this is an oviduct that has been stretched out. So it's obviously normally quite coiled within the female body. So fertilisation itself actually occurs high in the oviduct. In marsupials relative to placental mammals, the zona pellucida, which is the egg coating, is very much thinner and it's more easily soluble. So it's more easily to penetrate by acrosomal enzymes than in placental mammals. So when sperm and egg meet, the sperm undergo an acrosome reaction, which then causes the cascading effects that are required for fertilisation. So that results in changes of the egg coating structure and chemical signatures. So from studies in fat-tailed dunnuts, we know that sperm-egg fusion, so sort of during that storage in that region of the oviduct, that subpopulation of sperm that will move to the fertilisation site in the oviduct will actually have this kind of head rotation go on, where the sperm kind of form this weird T shape. And it's reported that sperm do not become hyperactivated during this transit period. So hyperactivation is a common form of motility in placental mammals, the sperm of placental mammals. So basically it's when the sperm become really agitated and beat really fast. So in fat-tailed dunnuts, we don't tend to see this change in motility pattern. But it's interesting that this seems to be different to what was recently found in the opossum sperm. So this was in the grey short-tailed opossum. A study reported that, in fact, the sperm of this species does become hyperactivated. So this is a very quick clip, but what we have here is uncapacitated paired and single sperm, and then capacitated paired and single sperm. So I'll just play it again. So I'll play it a couple of times so you can see what's going on and you can observe the difference in motility pattern. There you go. So that sort of more frenzied motility pattern is what we refer to as hyperactivity. So I'll drop this clip in the LMS folder so you can have another look later if you want. So sperm binding to the egg coat occurs by the way the acrosome reaction, and it then leads to the fusion between the contents of the egg and the outer acrosomal membrane. So penetration through the egg coating is facilitated by pressure exerted by the sperm head, and then passage of the sperm through the zona matrix is facilitated by this sort of dissolution of the zona material close to the inner acrosomal membrane. So females have been obtained where either the sperm head or tail is present within the zona pellucida. So this either indicates that sperm, so this can indicate that sperm may sometimes become partly stuck within the zona pellucida, or that the passage through the zona pellucida occurs relatively slowly compared with our placental mammals. And so you can kind of see here in this image the sperm of a diapausing tamer wallaby blastocyst, and there's a thickened shell coat and a compressed mucoid coat here with several trapped sperm tails. So yes, it's thought that perhaps that passage of the sperm through the outer coating of the egg occurs at quite a slow rate compared with placental mammals. So early development, after fertilisation, they undergo a period of conceptus expansion prior to blastocyst formation. So the total volume of the blastocyst is larger than the original egg volume. So this is different to what we see in birds and reptiles. So in marsupials, we see this non-rigid shell that stretches to accommodate conceptus expansion, following which this non-rigid shell will break to allow that placental attachment to the transient placenta. And this contrasts to what happens in monotremes. So in monotremes, there's a thick, rigid shell that is laid down to prepare for oviposition. So marsupials do have this transient yolk sac placenta, as it's called. So the embryo will first be encapsulated by shell membranes and floating free in the uterine for several days, at which stage it will hatch and then erode a shallow depression in the uterine wall. So it will absorb some nutrients from the placenta, but it's not this form of true implantation that we see in placental mammals. There's a very short gestation period, and then after birth, the embryo crawls from the cloaca to the pouch and attaches to the nipple. So in summary, marsupial development is characterised by early birth followed by prolonged lactation period. And so there is something very cool about newborn marsupial joeys that I will show you now in this clip. So it's to do with this sort of early coordinated movement that they have that allows them to travel from the urogenital sinus up inside the pouch.
Ultrasound shows the fetus repeatedly thrusting and grasping its forelimbs in a coordinated effort to prepare for its journey from the birth canal to the pouch.
Wallabies, when they're born, are extremely small, undeveloped, like an early human fetus. And even though they are born so small and immature, they can climb by themselves from the birth canal up to the pouch and attach their teeth. And they do it with this lovely Australian crawl movement. What we found in this study, quite to our surprise, is that that movement starts 3 days before birth. And they're really very, very fully developed. The brain is hardly organized. And yet they've got the coordination to organize those crawling movements. In humans, coordinated movements start about 12 weeks of pregnancy. Our wallabies, at the time they start doing their Australian crawl, are about 4 or 5 weeks equivalent developmental stage in the human.
So marsupials do lots of amazing things and lots of amazing reproductive things. One of which is embryonic diapause. So basically this describes a period of developmental arrest, and lots of the macropodids do this. So the red kangaroo is kind of like the classic example, and a description of what occurs in this species is that there'll be an initial pregnancy, say, that begins with a 33-day gestation, and then once this embryo is lactating in the pouch, the mother then becomes pregnant again immediately. So the presence of this suckling young arrests the development of that new embryo at approximately the 100-cell stage. So that specific arrest of the development of the embryo is what we call embryonic diapause. Diapause will then last 235 days, which coincides with the duration of lactation. So when the joey leaves the pouch, that uterine embryo resumes development and will be born approximately a month later. So the female then becomes pregnant again, but development is arrested. So there can be this continual process of the female producing young and lactating young and having one ready to go. So if you look at this in a diagrammatic way, basically this is what you have. So a mother can have three offspring on the go. Two offspring on the go. So you have the birth of, say, the first joey, after which the mother becomes pregnant again immediately. And then there's that period of, there's developmental arrest. So diapause occurs for that new embryo, a period of lactation. Then once that ends, true gestation begins again, and the joey is born and is suckling for 235 days. But of course, at that stage, the female also has another diapausing embryo. So just like this continual rollover, already having that embryo in a state of arrest and ready to begin developing as soon as the mother's not under the demands of lactating. So it is an arid adaptation that we see, as I said, in most of the macropodids, which are mostly continuous breeders. So you actually get this weird thing called concurrent asynchronous lactation. So this is when females can actually have different mammary glands producing different types of milk. So they will have a different they'll have different milk that's more nutrient rich and nourishing for the newborn young versus what older young might be lactating on. And so you can see here in this diagram how it can be adaptive to the arid environment, and especially when extreme conditions such as drought occurs. So here you have now let me just get this right. So this is mating, I think. No, this is mating. Oh. Anyway, this is a period of diapause along here. So the female has one young and another one, I assume that may be mating, is in diapause, in arrest. The first offspring is successfully weaned, but then there's a period of drought. And so that second embryo doesn't begin developing because conditions aren't right for the female. And so what you see is then a loss of young during that time, which is represented by the cross. Females can then even go into a period of anestrus, so they won't be in breeding condition when things are dire enough. But once conditions improve and the drought ends, estrus will begin again. Female will mate and give birth, and then the cycle continues again. So I guess the take-home message of this is the fact that during that period of extreme drought, the female loses young, but the investment at that point in time hasn't been too much, and so it's not such a huge cost to the female. So that's what marsupials do, but we do have many different species of native rodents in Australia, which are obviously placental mammals. And so they have a different way that they've sort of adapted and able to reproduce in arid conditions. And so just quickly, because we're not here to talk about placentals, but this is where some of my own research comes in. So basically we recognise that across Australia there's many different climate regions, so ranging from tropical rainforest up the top here through to arid desert in the centre. And then we find that our rodents do actually occupy basically nearly all of these niches. So obviously different species are found in different areas, but there's not much of Australia where you don't find a native rodent. And so what we're interested in looking in is how sociality may have contributed to, how sociality evolved in relation to these arid conditions. Because what you'll see here coded as the red species are our social species, and then the green species are our non-social species. And so when we looked at this, we thought that most of the social species are obviously occurring in the arid region. So that was a pattern that was quite obvious. We're interested in looking at whether that sociality evolved prior to occupation of the arid region, or it evolved outside of the arid region, and that allowed those species to then invade the those arid regions of Australia. And so we did what's called an evolutionary transitions analysis. I'm not going to go into this now, but it basically involved collecting climatic data from the rodents, each species rodents distribution over many years, and then looking at running this sort of evolutionary transitions, which was phylogenetically controlled, as to whether those species and non-social and social species, how that pattern influenced the evolution of sociality. So basically what we found is that non-social species in benign conditions remained non-social. However, we saw that non-social species moved into the arid region, and then under those conditions, those arid conditions, that's where sociality evolved. And then we did see some movement of social species back into the less harsh conditions. So in this way, placental Australian mammals have adopted a behavioural strategy of being able to reproduce in arid conditions, because sociality basically facilitates reproduction for a dominant breeding pair, and then other individuals assist that breeding pair. So that's sort of a reproductive strategy that has evolved in rodents, whereas in the marsupials we see that they have more of an independent strategy of embryonic diapause. So which strategy is superior in relation to how marsupials do it and how placentals do it? And when I say how placentals do it, I don't mean behaviourally, like I've just described in the rodents, but I just mean in terms of a prolonged gestation versus the marsupial strategy of a short gestation and the prolonged lactation period. So the traditional view just generally on marsupials is that they're inferior to placentals. They have low species diversity, they have a small geographic range, and there is this sort of tendency that they can be displaced by introduced placentals. And above and beyond that, people sort of think of their reproductive strategy maybe as being a bit weird or a bit primitive. So who does it better, marsupials or placentals? So we'll just have a quick look at some comparative developmental timelines. We all know how monotremes, marsupials and placentals reproduce, I hope, but this just demonstrates that. So just sticking to marsupials and placentals, obviously marsupials have this short gestation period followed by a long lactation period, whereas in placentals the gestation and lactation periods are typically more closely related in length. And what you tend to see overall is kind of like the young are weaned and independent of the mother prior to what you would see in a marsupial. So on that basis, you might think, okay, then the placental strategy is superior because the mothers are independent of the young a lot sooner on. It may have something to do with habitat though. So you might want to consider ecological differences between placentals and marsupials. But if you do that, at least in these couple of cases here, you generally see the same pattern. So this is the southern grasshopper mouse, and this is our familiar fat-tailed dunnart. And so you can see on this figure, in these sort of ecologically compatible species and compatibly sized species, the placental young are independent a lot earlier than the marsupial young, and that's the same for slightly larger species, the Thompson's gazelle and the Euro. So just a note on offspring size relative to maternal size. So the size of offspring in mammals typically depends on how sufficient they need to be at birth. So what we're talking about here is altricial versus precocial young. So altricial is very dependent on the mother, are basically helpless without them, won't survive without them. And if we look at this figure here, you might want to contrast the giraffe, which have precocial young. So obviously giraffes need to get up and start walking around within a few hours of birth to avoid predators. So if we compare a giraffe young with a human young over here, humans are fairly altricial. So the size of the young relative to the maternal size is a lot, the proportion is a lot smaller in altricial young. So they tend to be born at a smaller size and less well developed and equipped. But I really like this figure for the fact that you have the red kangaroo here. So the young are born at 1/100,000th of the size of the mother, which is just extraordinary. Looking at then comparative reproductive rates. So despite these very different life histories, the scaling of production rates is actually statistically indistinguishable across mammalian clades. So this study sort of demonstrates that. It used a life history database of over 4,000 mammal species. So this included marsupials and placentals. And what you will see first up is that maternal body mass relative to gestation duration differs between marsupials, which are coded in black, and placental mammals here. So as maternal body mass increases in placentals, gestation duration increases at quite a significant rate, but it's a lot reduced in marsupials. So the same kind of direction, but not significantly so. But what this study also explored is that maternal body mass in relation to lifetime fecundity didn't differ. So there's no difference in lifetime fecundity between marsupial species and then placental species. So essentially, this research showed that despite these kind of quite disparate reproductive strategies, females of the same body size all allocate energy to reproduction at essentially the same rate. And so this strikingly sort of similar results here on this side. They suggest that these evolutionary pathways that have led to these different mammalian physiologies and reproductive ecologies are in fact metabolically equivalent ways of being a mammal. So then if we go back to our question of what works best, well, marsupial and placental lineages produce biomass at equivalent rates. So it seems that traditional interpretation that the marsupial strategy is primitive to Eutherian strategy is a bit too simplistic. And then these different strategies may reflect adaptations to different environments, or they've just adapted in different ways to persist in the same environment. So as we said, these marsupials, they're investing less in newborn young, so there's less energetic cost when they are lost. And then pouch rearing and embryonic diapause is obviously very beneficial in the unpredicted and arid Australian environment. So I think the take-home message is that these two reproductive patterns are two independently successful reproductive methods. So just to finish up, I just wanted to cover seminal parity. So we've already learned a little bit about this, and most of what we do know is being from studies of Antechinus and Phascogale marsupials. So in all the well-studied species so far, it seems that females have a single highly synchronized estrus each year, during which males basically commit their lives to mating. So males die very quickly after this short breeding season. And in some populations, all the males are dead before the females give birth even. We learned in a previous lecture that the most common pathology is gastrointestinal hemorrhaging, which doesn't sound like a very nice way to go. And so what it seems is that endocrinological and pathological studies have shown that there's failure of the immune system and inflammatory systems, which eventually lead to this ultimate death. So why does this occur? What's seminal parity about, and is it adaptive? So apparently it is. It's suggested that it's an adaptive elevation of free cortisol concentration. And there's been this adaptive stress hypothesis proposed to explain it, which suggests that stress-related endocrine changes function to provide a short-term energy boost, which could be considered to be adaptive. So it's believed that there's adaptive advantages of elevating cortisol levels, because that then promotes protein catabolism. And you can see how this would allow small mammals with a limited ability to store energy as fat to sustain mating effort when forced to mate in winter. So if we just flick back here, this is the sex ratio of a population of red-tailed phascogale in the wheat belt. And you will see here that this significant drop here follows the breeding period in July. So the species that show seminal parity do tend to breed over winter. So it's interesting though, because males also sort of begin to disperse after mating, so they're getting a bit skanky, for better choice of word, a bit dishevelled and on their way to death. So they mate, there's an elevation in cortisol, but then females get really aggressive towards males as well. And so males are kind of then pushed out from the area and then they'll die. So it's possible that there's this additional adaptive benefit of removing males from an area to mean that there'll be more resources for gestating mothers and their young. So just another little peculiar and interesting reproductive strategy that we see in marsupials. So that's where I'm going to finish up, and there is a list of reading for further learning that you can take a look at. And I also wanted to point out this book, which I really highly recommend, Platypus Matters, The Extraordinary Story of Australian Mammals. So it covers all things on native Australian mammals, but it stems from Jack Ashby's obsession with platypuses or platypi. But it does talk about marsupials a lot, as well as other monotremes. And it covers a lot about the sort of historical opinions formed about Australian marsupials. And you can really see how many of these attitudes back then probably still shape our current, some people's current attitudes today, and may have influenced conservation issues surrounding our native mammals. So definitely give that a read if you like reading about science in your spare time. It's also very entertaining. Thanks for tuning in.